All right, guys, let's try this again. Welcome to week 8, lecture 1. The normal PowerPoint caption that I've been normally using for this has not been working. So we're going to play around to see whether Zoom captioning works. If all else fails, there's always YouTube. With that, we're going to go ahead and wrap up the rest of the Protists today. General announcement, we do have upcoming two exams within 1 week. So we have our laboratory exam number 2 covering the materials from weeks 4 through 8 on Tuesday, October 28th. And then on Thursday will be our lecture exam number 3. And remember, the day of the lecture exam is when the review assignment is due. And review assignment number 3 has slightly different questions for the extra credit part. So go ahead and take a look. Right after that, we have our November 1st Saturday point race field trip for both sections. So pay attention in lab for more information regarding when to meet. And for this one, we will be providing transportation. So we will be meeting on campus. And then the bus will take us to point race and take us back. Excuse me? And then remember that we do have a holiday, Veterans Day on November 11th. We do not have class on that day. November 18th is the last day to drop this class with a W, and then our lab exam number 3. I think this is our final lab exam for this course, will take place on November 20th. With that, let's go ahead and do a quick knowledge review, and that is the diatoms are distinguishable for other members of straminophiles by which of the following? The fructules, chlorophyll A, chlorophyll B, flagella, or none of the above. So which of these traits make diatom unique among the straminophiles? Go ahead and pause the video and then we'll go over the answer. All right, so the thing that makes diatom unique are their shells, their cell wall with silica, and that is called fructules. So it is choice A. Today's lecture is still on Protists. We're going to try to finish up the rest of the Protists today, and start by continuing with the straminophiles group, supergroup, and then we're going to go down the list from brown algae, red algae to green algae, and then wrap up by talking about the slime modes, which, despite of the confusion with the name, is a protist and not a fungi. So remember again that as a group we call Protists is paraphyletic. It is all the branches that's in yellow, but because it does not include the branch that includes our current modern day land plants, the fungi, and the animal grouping, it is paraphyletic. And for today, when we're talking about brown algae, which is right here, red algae, which is right here, and green algae, which is our closest relative to land plants, you'll note that they come from different branches, and so the grouping we call algae is also paraphyletic. So there is a lot of things that we use to group together based on their morphology and the habit of living, that once we study them in more depth with the invention of microscope and molecular technology, then it becomes clear to us that these are convergent evolutions, and based on appearance and where and how they live alone is not an accurate way to group things. We're going to start by talking about brown algae today. So again, we're still in the straminophiles. And algae, any of you guys ever seen kelp know that they are very much multicellular. The kelp forest can get very, very large. And the kelp are, of course, benthic. That means that they are attached to something, a substrate such as the ocean floor, rocks, for example. And they are marine, so they live in the ocean. Brown algae tend to prefer the cooler waters. And even though we will focus on brown algae just because they're so important economically and environmentally, there are more forms of brown algae than just kelp. So you can see them in filament as well, I think, for some of the examples we have in lab. Brown algae have chlorophyll A, chlorophyll C. Note that, remember, cyanobacteria has the oldest form chlorophyll A. So now we're at brown algae, there's also another version called chlorophyll C. And brown algae do also have carotenoid. They specifically have a type of carotenoid called fucoxanthin. Something that's interesting to note for these is that they do have not rigid cell walls that contain cellulose. And in addition to cellulose, they also contain mucilage. So this is like the sticky, viscous, sort of snot-like substance. And it contains two molecules, algin and fucoidin. Specifically, though, I want to draw everyone's attention to the fact that their cell wall does contain cellulose, just like land plants. And for that reason, there's a lot of folks who study algae who are, yes, in the department of plant biology because a lot of the algae grow very quickly. They're fairly small in size and they're simple to grow in the lab. So if you are interested in studying cell wall, especially plant cell wall, algae makes a really great model organism because of cellulose. Now we're going to talk a little bit about the two types of chlorophyll. So you guys are not expected to memorize these structure. You do not need to memorize these, but just so you have the idea in your mind that for both chlorophyll A and C, the porphyryl ring is still very much intact. You still have your nice magnesium ion sitting in the middle and its interaction with the nitrogen around it. However, what is different between chlorophyll A and B versus chlorophyll C? So chlorophyll A and B is what you see in the land plants, is the different side group chain modifications. So A and B differ on reaction group number 7 here, and chlorophyll C we're at reaction group 8. And this is because chlorophyll C is something that involved in the evolution up to the diatoms and the brown algae, along with the synthesis of the carotenoid that's called fucoxanthin. So this is something that is unique or a synapomorphy of the branch that includes diatoms and brown algae. And here's just a reminder that there's more than kelp to brown algae. Here is another non-kelp brown algae, genus Laminaria. Very cool name there. It is not the kelp we're used to seeing in the giant kelp forest, but you can see that it does have the typical body plan you would associate with a kelp forest that is common for this genus, and that is you would have something that's called a holdfast. So note that for all that some of the brown algae can look very similar in their body plants to land plants, they do not have roots. They have a holdfast. There is no structure that penetrates into the substrate beneath it. The holdfast purpose is to attach the other two parts to the substrate, such as the ocean floor. Above the holdfast, attaching it to the rest of the structure, usually the main structure used for photosynthesis. In this case, is a blade. So blade, you'll note, is the common term we use for anything that's flattened and vegetative for photosynthesis. The brown algae often will have an air bladder. So in this case, it's usually located at the base of the blades. So this helps the algae stay afloat so its blades can spread out and catch light. And the stipe is what connects the blade and the bladder to the holdfast. What is also interesting about brown algae once you get to laminaria is see this thing, right? See how that looks awfully familiar, right? Hopefully you guys will remember and notice that this look very familiar to the seed plates in the seed tube elements of land plants. So they do have transportation tissue or the more ancestral form of it in that they do have something that look awfully like seed tube elements in the phloem of land plants. However, they do not have true vascular tissue because they do not have xylem and they do not have lignin. And Professor Oliver poses this question to you guys on why do kelp not need xylem? So go ahead and pause the video if you like and think about it for a moment. And the answer for that is, of course, because kelp is surrounded by water. So you do not have to worry about all the support and transportation you need to move water against gravity up to the chute because the chute-like structure, in this case, is surrounded in water. Everything is surrounded in water, so you don't have to worry about transporting water against gravity. Now we're going to talk about the life cycle of the, well, here it says laminaria, but this is something pretty common for brown algae, and that is sporic meiosis. If you have any questions about this part, make sure you ask because sporic meiosis and all of these terms are going to haunt you from protists all the way until ferns, so the seedless vascular plants. So make sure you're clear about the ploidy and the name and the function. So sporic meiosis is where we have something that's also known as the alternation of generations, where you alternate between two clearly visible forms of the plant to complete one life cycle. You have to alternate between the 2N phase, the diploid phase, and the N or haploid phase to complete the life cycle. So that's why it's called alternation of generations, because you are alternating between diploid and haploid and diploid and haploid and diploid and haploid. It's also known as sporic meiosis because you make spores. Specifically, you start off with a structure that's called the sporophyte, which is diploid. And on the sporophyte, you'll have the structure called sporangia, which is where meiosis occurs to produce spores. In this case, the spores are called zoospores, and they are mobile and can swim. The spores, in this case, zoospores, will germinate, just like the spores we have in the ferns will germinate in lab, and they will germinate and produce gametophytes. Because meiosis has happened and the spores are haploid, or N, the gametophytes that they give rise to is also haploid and have a ploidy of N. And then after that, the sperm will swim to the egg, fertilization will occur, and you'll have restoration to diploid or 2N in the formation of a zygote. So once you have fertilization, you have a zygote, which is diploid again, and then the zygote can grow and do mitosis and become a large multicellular organism that we call the sporophyte. And then the sporophyte can make sporangia, where mitosis occurs to make more zoospores, and then the zoospore can then give rise to gametophytes, which produces gamete, and then the cycle continues. So again, make sure you are clear on the life cycle of these things, specifically the terms like sporangia, sporophyte, spore, gametophyte, gamete, fertilization, and zygote. These are really important for a lot of the family we're going to be covering in the next few lectures. And please, when you're memorizing these vocabulary words for the different structures and forms also keep track of the ploidy count. So sporophyte and sporangia is 2N or diploid. Once you have meiosis and make the spore is haploid. The gametophyte is haploid. The gamete is haploid. The zygote is diploid. And then again, we go back to the sporophyte, which is diploid. So please, please, please keep track of that. All right, now we get to the giant kelp that everyone has probably seen at some point, given that we're in California and these sort of things just show up in our culture. The giant kelp, as the name implies, is very large. It is the dominant life form of kelp forests, which is very important for this specific ecological environment because they are very productive. They're as a primary producer, they provide a unique habitat for many, many, many species. And unfortunately, they are under threat by climate change because remember, brown algae prefer cooler water, so the increasing temperature has not been great for it. And also, as a result of environmental change due to human activities, there has been an introduction of a lot of invasive species as well that is really decimating the giant kelp forest. So we do need a lot of restoration ecologists and a lot of researchers in this area to keep and save our giant kelp forests off the coast of California. Here is another type of brown algae, the sea palm, Postelsia. It looks very much like a palm tree. You have a stalk and you have your leaves, so-called leaves, on top. And in the case, this is not a true leaf, so it is a blade. Yet another type, in this case, the fucus is not a kelp, but you can still see the air bladders. In this case, it's not located at the base, but it is embedded in the blade. Another type, Salgalsum. So you can see in this case, we do not have the giant structure with the stipe attaching the blade and the air bladder to the floor of the ocean. However, you can see the air bladder at the base of each blade still along this entire stipe. And they're often found floating in large mats on the east coast as part of the Atlantic Ocean. And brown algae in general is very important not only for its ecological uses, but for its economic uses because it contains algin as part of the cell wall. So this is a great stabilizer for different types of solutes, and it's an emulsifier so that, when you have something that contains things like dairy, things don't just separate whenever you freeze it. So algin is an additive for a lot of things that contain dairy and various other substances. Next time you eat ice cream, read the ingredient. A lot of them will have algin. And also, whole algae is sometimes just eaten, especially if you're like me from East Asia. We have kelp salad, which is delicious. So, economically important. Now for a knowledge check. So the two types of chlorophyll that brown algae have are which two types? Go ahead and pause the video and then we'll go over the answer. So brown algae have chlorophyll A, which is the more ancestral form that's also found in cyanobacteria, and chlorophyll C, which is something that involved in this particular branch that it shares, brown algae shares with the diatoms, which means diatoms also have chlorophyll A and C. Okay, now we're going to switch gears a little bit to talk about a different branch. So the last lecture and today's lecture, we're talking about the shrub monophyles, which is this particular branch. Here are the diatoms, here's brown algae, and then woolmycetes. Now we are going to move and talk about this branch. There are Archaeplastida, and it's also called the Plantae supergroup because remember, this is the group that includes all land plants that are still living today. And students tend to prefer the term Plantae supergroup for obvious reasons, because it's shorter and easier to spell and pronounce. I will accept either Plantae or Archaeplastida on exam. If you recall that strepmenopiles, the synapomorphy for those are the two flagella, right? One hairy and one smooth. Even though for some of the groups you might not be able to visibly see them, like brown algae. If you think about giant kelp, you usually do not think about the hairy flagella and the smooth flagella. They still have the protein for those flagella, so that is why it's still a synapomorphy. In this case, for the Plantae supergroup, the synapomorphy is chloroplast from primary endosymbiosis of cyanobacteria. In other words, if you study the molecular evidence of the chloroplast for everything in this branch, the Plantae branch, you'll find out that they share a common lineage as the cyanobacteria. In other words, when this entire branch branched off from everything else, somewhere over here, ancestral line engulfed a cyanobacteria and performed endosymbiosis and kept that cyanobacteria around. And over the course of time, that cyanobacteria that got engulfed became the chloroplasts that we see in the land plants all around us. So for this branch, there are several notable groups: the glaucophyte, red algae, green algae, and land plant. And we're going to start by talking about the red algae, which is also commonly called rhodophyta. So rhodo means red, like rhodamine, which is a red dye. Red algae are also benthic and marine, just like the brown algae. However, they tend to prefer warmer water compared to brown algae. Remember, brown algae prefers cooler water. They are also found in various body forms. They can be unicellular or filamentous or have large three-dimensional structures. So even for the large three-dimensional structures, they tend to be smaller than brown algae. Remember that chlorophyll C is something that involved for the brown algae and diatoms family, right? So we do not have chlorophyll C for the red algae. We have instead chlorophyll A, so we kept the ancestral form. We have carotenoids in red algae, and we have the red pigment called phycourizoin. So urizoin means red. Hopefully, it will remind the medical people among our audience of erythrocytes or red blood cells. Phycourizoin, phyco is usually the prefix that means plant, so plant's red color, in other words. Phycourizoin is one of the phycobilins, so it is one of the accessory pigments that you find everywhere, and I believe you see this in no, you do not see this phycourizoin in cyanobacteria, though cyanobacteria does have phycobilin. So that sounded a little bit confusing. Let me rephrase that. There are many types of phycobilin, and phycourizoin is one type of phycobilin that you see in red algae, and it helps collect the different wavelength of light than chlorophyll, but it will reflect red light, which is what gives it its reddish color. However, red algae is not always red and green algae is not always green. So like keep that in mind. It just means that it does contain this molecule. It does not mean that it is the dominant pigment. Okay, red algae do have cellulose and mucilage as well, but in this case, the mucilage has agar and carrageenan, which I have heard pronounced several ways, so be prepared for different pronunciations. And these can grow up in much deeper water compared to brown algae. They do have a more complicated sporic meiosis level, so there is an actual third stage, but don't worry about that for this class. If you decide to go and specifically study algae for your living, then you can worry about that. One type of red algae that's really cool is coralline algae because these are algae that have a rigid cell wall. So remember, for red algae and brown algae, they have cellulose and mucilage as their cell wall, so it's usually flexible and not rigid, right? Well, here's our exception. They have a rigid cell wall because they deposit calcium carbonate in their cell wall. And that makes them really hard, really, and they are important for the formation of coral reefs because they help glue together the coral reefs essentially. And so their presence is important for coral reef health and productivity. And remember, corals are animals. So they look like corals, but coralline algae is a type of protist and not an animal. Red algae can survive at greater depths than other algae because they look red. They don't always look red. Correction. They don't always look red, but it's because they have phycoerythrin. And the phycoerythrin molecule looks red and absorbs blue light, and blue light can penetrate the ocean to much greater depths than red eye or green light. Because if you review, I don't know what physics class you guys took, but for the level of electromagnetic radiation, the violet end of the light tend to have shorter wavelengths at higher energy. And higher energy means greater penetration, whether it's penetration of tissue or penetration of water. So if it can absorb blue light, that means it can still capture light at a lower depth in the ocean compared to something that can only absorb green light, in which case green light does not penetrate as well through the layers of ocean water. And so those things can only grow at a shallower water. And especially considering chlorophyll mostly absorbs a lot of red light. One's peak is red, the other one is blue. For things that mostly capture red light, you can only grow fairly close to the coast. Again, I was not very clear about this, so I want to emphasize phycoerythrin looks red and absorbs blue. Red algae has phycoerythrin, so it can absorb blue light and grow at greater depths. But red algae does not always look red because even though it contains phycoerythrin, phycoerythrin may not be the dominant pigment in there, so it may not always look red. Okay. All right. So red algae can be useful to humans because agar and carrageenan. So if you look at this structure, can you guess what they are based on these? Hopefully everyone who has studied glycolysis will be able to recognize these because anytime you see this little, I don't know what you call this shape. I think one of my friends usually calls them the lawn chair shape. Anytime you see these, I guess, very sci-fi looking lawn chairs, that means you have polysaccharide. So you have a 6-ring sugar. So that means that red algae cell wall has cellulose and additional types of polysaccharide in the form of agarose and carrageenan. And yes, this is the agarose you use for your gel electrophoresis in the molecular biology labs. And some of it is, I think, what you use for certain types of snack as well. So that brings us to the economic use. So porphyria is also known as nori for those of us from East Asia. It is used in sushi, and if you ever eat wonton soup, it's commonly you put it in there as well. Agar acarnate are used as emulsifiers in many types of food. And agar is one of our key things we use in science. So it's the gel for electrophoresis. I forgot to mention earlier, but it is the gel for the gel plates for your bacteria culture and also our fern cultures that we're growing. So there's extract of red algae in those things. While I was putting together the slides and looking them over, I got curious about whether or not red algae, which prefers warmer water, is affected by climate change and how it will respond in terms of the biomineralization or like the deposition of calcium carbonate on cell wall. So I was looking at paper on that out of curiosity. The short answer is yes, climate change still affects red algae. But one thing that came up by accident is I found out that apparently there's a lot of aquarium stores that sells coralline algae in bottle. So this is something I found. This one is from Amazon, I believe, which is kind of hilarious and kind of cool. And one source of how you can find research material for certain things is just through Amazon. So another source of economic importance, in addition to the food and the lab stuff, is aquarium use. All right, now for our next knowledge check. Red algae can grow in greater depths of water because of what? Go ahead and pause the video and then we'll go over the answer. So red algae can grow in greater depths of water because they have phycoerythrin, which absorbs blue light, and blue light has more energy and can penetrate the ocean water to greater depths. For an exam, I would be looking for something specifically, the keyword I'm looking for is phycoerythrin. And if you mention something about, absorbing blue light or absorbing light at greater depths, that would work. Okay, now moving on to green algae. So we are here at this branch of the Plantae supergroup, and green algae are some of the closest living relatives to land plants. Specifically, there are two groups, Chorophytes and Charophytes. And unlike the brown algae and the red algae, green algae tends to be mostly freshwater. They do also come in various body forms, so they can be unicellular or they can be filamentous or multicellular and xalic. So xalic means that there's no clear differentiation of parts. They have what's different here is zygotic meiosis. So a few of them have sporic. So zygotic meiosis means that when they're doing the alternation or generation between haploid and diploid, the haploid form is the dominant form. For the sporic meiosis, which is the one that we've been previously talking about for brown algae and red algae, the sporic meiosis means that the diploid form is the dominant form. So for our land plant, the diploid form is the form that's dominant. It's the form that we see that gets so big. Green algae do have many similar traits to land plants. They have chlorophyll A and B. So chlorophyll B is green algae and land plants. They do have carotenoids, which tends to be show up in all algae. Specifically for cell wall, notice that their cell wall is made of cellulose, hemicellulose, and pectin. Looking awfully familiar there, right? Because these are the same structure or the same components you see in primary cell wall in land plants. Green algae do store energy as starch in plastids, which is something that land plants do. If you recall back to when you're looking at potatoes in the lab and sectioning them, and you see all the amyloplasts, like tiny round crystals, right? So green algae can do that too. And some of green algae can actually make phytochrome. So in the more recent lecture, remember phytochrome is the sensory pigment that is critical for many of the plant's response to light, such as shade avoidance and circadian rhythm. So that already has evolved with the green algae. Here's a reminder from Professor Oliver as well. The colors do not correlate with the name. So here is a red algae that looks green. It is a freshwater red algae. Here is a green algae that looks red. So red snow, I think some people call it like watermelon snow because it looks pink. Sounds very delicious, but it is a green algae. And so ultimately, the color you see for either these or even the leaves in land plant, depending on which pigment it has more. Even though it looks green here, it probably still has phycoerythrin, just not the dominant form. And even though it looks red or pink here, there is still green pigment and chlorophyll in there, it's just not the dominant form. So again, the red and algae name by itself can be kind of misleading, but you are expected to know that red algae has phycoerythrin and green algae has chlorophyll A and chlorophyll B. Oh, here's green snow. So you have red and green. That's very Christmassy. But all right, so these, I forgot to mention that these are algae that can grow on the snow, which is a type of adaptation for very low temperature and relatively nutrient poor, depending on the age of the snow. Green algae. Green algae. Here are the two main classes. The chlorophytes and charophytes specifically belong to the Chlorophyceae and Charophyceae. So the CEAE name is also something that you commonly see in the family name of land plants as well. If that helps you remember when you see this name, that these are the things that are more like land plants. Chlorophyceae is the branch that's less like land plants, and Charophyceae is the family that's more like land plants, and it probably contains the ancestors, excuse me, to land plants. And the reason we think that is we looked at how the cells divide, or the mode of cytokinesis, the symmetry and the location of the flagella, as well as their mechanism for deposition of cellulose. Specifically, cytokinesis and cellulose deposition are one of the reasons why we think Charophyceae might be the closest lineage to land plants. Now, what do we mean by when we say the methods of cytokinesis? Well, Chirophyceae have something that's called a phycoplast, where when they are depositing the new cell wall that separates the daughter cells, the microtubules that are laid down that guides the deposition of the new cell wall are parallel to the new cell wall that's going to form. In contrast, Charophyceae and all land plants have something that's called a phragmoplast, where the microtubule that's guiding the deposition of the new cell wall is perpendicular to the new cell wall that's going to form. So these are parallel to the mitotic spindle, but again, perpendicular to the new cell wall. And just to refresh everyone's memory, here's a video or animation of phragmoplasts and cell division in the land plants made by my former lab mates. Oh, the sound for this is terrible. Okay, so here is the formation of the phragmoplast. Here, the small things are the Golgi. Golgi vesicles are being deposited as the new cell wall is formed. Here are the two vacuoles that are forming as well. In contrast to the animal cells, plant cells do tend to have a lot more Golgi. Here are the microtubules that guide the deposition of the vesicles from the Golgi to the new forming cell wall. And when they first form, they form a fenestrated sheet, which has holes in it, but as the deposition of the vesicles continue, they will eventually fuse and flatten out, and that will form the new cell wall. And there you have it. Now you have two daughter cells. The second thing that separates the two family of chlorophytes and charophytes are the location of the flagella. The chlorophytes are in the family that includes the chlamydomonas. So previously I mentioned that some people study algae in the Department of Plant Biology because they have cellulose, right? So you study cell wall, algae makes a great model organism. I know one professor at UCLA who studies brown algae. I know so many professors who study green algae because they're closer to the land plants. And Chlamydomonas is one of the favorite molecular models for green algae because we do have genetic tools for them. And they are mobile, so they do have apical flagella and they have symmetrical cells. In contrast to this, the charophytes have a subapical flagella that are located elsewhere, and the cells are asymmetrical. And the third thing is that cellulose microfibril deposition are deposited by rosettes only in charophytes. So this is the family that is closer to land plants. And if you recall from way back week 2, the deposition of cellulose in land plant by cellulose synthase involves the formation of cellulose synthase into rosettes. Well, chlorophytes of green algae can also do that. They have a circular enzyme complex that secretes cellulose microfibrils outside of the plasma membrane. So that's another piece of evidence indicating that they are probably more closely related to land plants. The green algae and specifically chlamys come from the volvocene line, so these can be unicellular or multicellular or colonial. Chlamys, again, is one of the popular lab organisms. They're very small, they reproduce very quickly. They have the same type of chlorophylls as land plants, and they have the same type of cell wall and the same mechanism for cellulose deposition as the land plants. They do have a contractile vacuole, an eye spot, a nucellus, and a paranoid to help concentrate the CO2. And here's what they look like under a microscope. So hopefully you guys will be able to see this in lab. Why is this sound working so well? So you can see here are the two flagella right here. They do actually have oil bodies as well. And chlamys are mobile. You can see that one went by pretty quickly. And when they divide, they move slightly slower. So look forward to these in the lab. And a lot of the drugs that you can use to study cell division in the plants will also have an effect on chlamys. So that's what's nice about them. And because for their effect, a lot of the effect means that they don't move well anymore. That makes the symptoms really easy to spot. And chlamys are one of the green algae that have a zygotic meiosis. So remember, zygotic meiosis is the alternation of generation, but its haploid form is dominant. And in contrast to the sporic meiosis, zygotic meiosis means that soon after fertilization, you form a zygote, and then you form a zygotic spore, a zygospore, and then meiosis happen right then. So you don't have sporangia with the formation of spore and the spores going through meiosis. You have meiosis happening at the zygote part. So that's why it's called zygotic meiosis. And as you might imagine, the reason why sporic meiosis is called sporic meiosis is because meiosis happens at the spore cell. So in this case, for chlamys, you have meiosis happening at the zygospore, and then you can have the germination of two lines that are haploid, the positive string and the negative string. And once these two strings grow up, they will give rise to zoospores to produce gametes, and then the gametes will fuse. And you have fertilization, and then you have zygote, and then you have meiosis again at the zygote stage. So that makes them different because meiosis is not happening right here, right before zoospores. It is happening right here, right after zygospores. Here's what the other members of the volvocine line can look like. So you have colonies of A232 cells forming these little balls in Pandorina. You have volvox, which looks very cool because they have these thousands of cell round colonies, and then their daughter colonies form inside the mother colony, and they form like small balls inside a big ball, and it looks very cool under the microscope. We also have Spirogyra. So Spirogyra is a chirophyte, and they do also do zygotic meiosis. So they have conjugation between the positive string and the minus string, and then here you can see the formation of the zygospore. And I believe we should be able to get live samples of these in lab as well. These are filamentous, by the way, in contrast to the colonial and the multicellular ones. The last form we want to talk about are the sea lettuce ova. It is a chlorophyte, so this is a branch that's slightly less related to land plants. Remember, the charophyte is the one that's more related. And the reason we want to mention the ova specifically is because in this case, they have an alternation of isomorphic generation. So this is halfway between zygotic meiosis and sporic meiosis. Because in this case, you have both haploid and diploid forms, and they're equally dominant. They look exactly the same. They have a zeolic body form, so there's no differentiation of like stipe and blade and bladder and the anchor. Everything just looks pretty much the same. And here are the haploid and diploid, and they look exactly the same. So that's pretty interesting. Now for our next knowledge check, how are green algae similar to plants, specifically to land plants? List two traits that make them similar. And there are more than two, so you can list as many as you want when you're reviewing. Go ahead and pause the video, and then we'll go over the answer. So there are four things you can mention here. One is that green algae have chlorophyll A and B, just like land plants. One is their cell wall composition is similar to land plants' primary cell walls, so they have cellulose, hemicellulose, and pectin. The third one is that they store energy as starch. And then the fourth one is, I'm blanking out for a moment here. Let's see, chloroplast, chlorophyll, excuse me. Chlorophyll, starch. Oh, and there's more than four. So phragmoplast, the charophytes specifically can do the phragmoplast division into daughter cells, just like land plants. And then there is also the rosette of cellulose photosynthase, forming a rosette and secreting microfibrils outside. So any of those would work. Oh, and then there is also that some of the green algae can make phytochrome. So any of those six things, just list two of them and it'll work. All right, now we're moving on to a different branch. So today we started off with the brown algae, finishing up the strepmonopause. Then we talked about the plantae supergroup, also known as Archaeplastida. And now we are going to move to the episthoconts. So we are here in the metazoa supergroup. Fungi are here. So we are, yes, more closely related to fungus than we are to plants. And then amoebozoa is the protists that we're going to be talking about. And you can see there was a reason why they tend to be clustered in this branch more closely with the fungi than with others. And part of the reason for this is they move by pseudopoda and they are non-photosynthetic. So these are heterotroph and they have to survive by digesting other things. And this amoebozoa includes slime molds and amoebas. And like water molds, despite of the word mold in the name, slime mold is a protist and not a fungi. The amoeba supergroup includes phylum Plasmodial slime molds, and they look something like this. There's a name specifically for this form called the Plasmodium stage, which is the feeding stage. So these are diploid. You can think of them sort of like the equivalent of sporophyte in being the diploid stage of the reproductive cycle. Please do not confuse Plasmodium with the Plasmodium, which causes malaria. And so it is unfortunate naming sometimes, but this is how it goes. And yes, capitalization is important, and so is italicization. But for the particular form of Plasmodium that we're talking about, Plasmodium is something that looks like this. It does look like fungi, and it also has a behavior that's very similar to fungi in that the Plasmodium is a giant single multi-nucleate cell. So fungi does this a lot with their aseptate hyphae, which we'll cover in the next lecture. But again, remember that this is the feeding stage of the Protists group of the amoebozoa supergroup. And for this, it is the feeding stage called the Plasmodium. And it is diploid. Again, remember to track the ploidy of these different stages because they're important. So here are how the reproduction of these group of slime mold works. So when the condition is favorable and there's plenty of food, it is in the plasmodium phase, which is 2N, which can then feed and digest things. Under unfavorable conditions, it will form sporangium. Plural form is sporangia. And that's what it looks like. It'll be a little cluster of things on a stalk. And this little cluster of things is the sporangiospore. So instead of the sporangium with the spores forming inside, you will be looking at the sporangiospore, which is, I guess, you can think of it like how you call sporangia in slime molds. It's called the sporangiospores. And similar to the other algae where you see sporangia, meiosis happens here and you form spore, which is haploid. So again, Plasmodium is diploid. Once you have sporangia, or in this case, sporangiophore, and you have meiosis, now everything after is haploid. So the spore is haploid, and the spore will germinate, and they will make gametes, which are amoeboid-like cells, right here. So again, very similar to algae, you have sporangium, you have meiosis, you have spores germinating, forming gametes. And the similarity continues. Then you have fertilization. Now we're back to diploid zygote to end. But what is different for the slime modes compared to the fungi is that when you have mitosis of the zygote, you don't have cytokinesis. This nuclei just keep dividing, making more nuclei, but there's no formation of new cell walls. So that's how you get this large cell that has many, many nuclei in them. That is the plasmodium. So this will reform the feeding plasmodium. And so to recap, you have the 2N plasmodium, which does meiosis inside the sporangia to form the spores, which is haploid, and it gives rise to haploid gamete, which then does fertilization to go back to your 2N feeding stage plasmodium. The sporangial 4 is the sporangium plus the stalk. And sporangial 4 will form when the condition is unfavorable because spores are super handy, so they can go dormant for a really long time. Some of the spores you have probably heard in the news can survive vacuum of space and the radiation of space. So you can imagine when the condition is not great, when it's too dry, there's not enough food, they can make spores, the spores are dormant, and they can wait until the condition gets better. In addition to the slime modes that we have just talked about, here are some other forms of slime modes. So chocolate tube slime, the Bacea sporangia, and then you have the nice sounding dog vomit slime mode. And you can see based on the photos why people used to think that they are a type of fungi and hence gave them the mode name. But again, these are a type of protists and not fungi. Uh, PBS did not allow me to embed the video, but there is a video here because slime mold is another type of popular model organism. They reproduce quickly, and they're small in size. They have a very easy to manipulate genome, so you can have molecular tool for them. And they do have a lot of interesting traits. They have tropism. They can do circadian rhythm. So there are a lot of labs that research slime mold as well. And you can go here and see how people use slime mold to navigate maze and make decisions. And it's a very cool video. So that pretty much wrap up today's lecture. We have covered up to amoebozoa. Next time, we're going to go and get into the fungi. Now is our last knowledge check. Name 2 processes that reduce the ploidy to haploid, which is A, and then increases it back to 2N, which is B. Go ahead and pause the video and then we'll go over the answer. So this is meiosis and fertilization. And on exam, I will probably not make it this simple. I'll probably ask you guys to remember a couple of the other stages as well. So sporangial 4, sporangium, gamete spore, zoospore, zygospore. Be aware of the name of all of those. The plasmodium versus the sporophyte. Be able to keep the ploidy through all of those. And if you have questions, as always, leave me a message, ask me in lab. And with that, I'll see you guys in lab. And the caption did not work for this entire thing, so we'll see how the caption for this goes. Thank you for your patience with the technology.